Welcome back to the science behind. Most people will judge a fish tank the same way that they will judge a glass of water. If it's clear, it feels right, but if it's cloudy, then something will feel off. And that reaction makes sense. Humans have always trusted clear water. Being able to see through it usually means that it's safe, and that instinct has carried straight into aquariums. So clear water has become the goal. We filter for it, we clean for it, and we measure success by how polished our tanks look. But an aquarium doesn't actually run on appearances. It runs on chemistry and biology, and those don't always line up with what we see through the glass. So in this episode, we're going to break that apart. First, we'll look at what clear water actually is, what it physically means, and what it doesn't tell you. Then we'll get into the chemistry, which you can't see at all, and the stuff that causes most tanks to crash without any warning. And after that, we'll talk about where the biology really lives in an aquarium and why some of the healthiest systems don't look clean at all. And finally, we'll look at the situations where crystal clear water is actually a warning sign and not a sign of success. Because if clear water really meant healthier water, aquariums would be simple. But they're not. When we describe aquarium water as clear, we're not describing its chemistry or its biology. We're describing how light behaves when it passes through it. Water clarity is governed by optics, specifically by how much incoming light is absorbed, scattered, or reflected before it reaches your eyes. In an aquarium, anything that floats freely in the water column and is large enough to interact with light will reduce clarity. Suspended algae cells, fine detritus, bacterial blooms, uneaten food broken down into microscopic particles. These particles scatter light in multiple directions, which is why cloudy water looks dull or milky instead of being transparent. Mechanical filtration works by removing exactly those particles. Sponge pores, filter flush, polishing pads, all of them are designed to trap matter within a certain size range. As particle concentration drops, light transmission improves and the water appears clearer. So, when a tank looks crystal clear, what that really tells you is this: there are very few light scattering particles suspended in the water column. And nothing more. Because most of the substances that define whether an aquarium is healthy don't interact with light at all. Ammonia exists as dissolved ions. It doesn't scatter any light. Nitrite and nitrate are fully dissolved salts, which means that they don't change transparency. Carbon dioxide, oxygen, and nitrogen are dissolved gases, completely invisible. pH is a measure of hydrogen ion concentration, which means it has no visual signature. You could double the ammonia concentration in a tank and the water would still look exactly the same. You could cut dissolved oxygen in half and see no change through the glass. From a physical standpoint, clarity is blind to chemistry. There's also an important scale issue here. Many biologically active molecules exist at concentrations measured in parts per million, or even parts per billion, and at those levels they simply do not interact with light in a way where the human eye can detect it. Our vision is good at spotting macroscopic particles. It is completely useless for detecting dissolved chemistry. This is why water treatment plants, laboratories, and aquaculture facilities never use visual clarity as a health metric. They measure chemistry directly because appearance is not predictive. In aquariums, though, we rely heavily on sight, and that leads to a false sense of security. Clear water feels controlled, it feels stable, but physically, all it means is that the water column is optically empty. It says nothing about what is dissolved inside it. And it says nothing about whether the system is actually functioning. That gap between what clarity shows and what it hides is where most aquarium failures begin. Once you do separate appearance from function, the next question becomes unavoidable. If clarity doesn't tell us whether water is healthy, then what does? The answer is chemistry, and most of it operates completely out of sight. At the center of aquarium chemistry is the nitrogen cycle. It's a constant, ongoing chemical process. Fish excrete ammonia directly through their gills. It's not waste that sinks or floats, it's dissolved immediately into the water. At very low concentrations, ammonia already becomes biologically active. It interferes with gill function, damages tissues, and disrupts ion regulation. And yet, visually, nothing will change. The water will stay clear, the tank will look fine, and in a stable system, ammonia is rapidly converted by nitrifying bacteria into nitrite. But nitrite introduces its own problems. It binds to hemoglobin and reduces the blood's ability to carry oxygen. Fish can be surrounded by oxygenated water and still experience internal hypoxia. Again, there is no visual sign, no cloudiness or no warning. Nitrite is then converted into nitrate. Less acutely toxic, but far from harmless. Elevated nitrate acts as a chronic stressor. It suppresses immune response, slows growth, and increases disease susceptibility over time. A tank with high nitrate can look pristine while slowly degrading the fish's health. This is where the idea of silent failure becomes very important, because many aquariums don't crash suddenly, they erode. Fish will survive, but under constant physiological stress that the keeper will actually never see. pH adds another layer of invisibility. Small shifts in acidity can change how toxic ammonia actually is, alter enzyme function, and stress fish long before their behavior will change. Buffering capacity determines how stable that pH really is, but alkalinity has no visual indication either. Temperature and oxygen interact in the same way. Warmer water holds less dissolved oxygen while metabolic demand increases. A tank can look calm and clear while oxygen availability will quietly drop below what the fish will require. This is why new tanks often fail despite looking pristine. The water is clear, the decor is clean, and nothing appears to be wrong, but the chemistry has not stabilized. The nitrogen cycle is not complete and the buffering is weak. Small changes therefore will have outsized effects. The same thing happens in heavily stocked tanks with a strong mechanical filtration. The water will be polished, debris will be removed, and visibility is high, but chemically, waste production can actually then exceed biological processing capacity. From the outside, both tanks will look healthy, but chemically they are not, and this is the core problems you find with judging aquariums by clarity. The most dangerous conditions don't announce themselves, they don't cloud the water or they don't tint it, they accumulate silently until biological limits are crossed. Most lethal aquarium failures are therefore not visible failures, they are the chemical ones. By this point of the video, one thing should be very clear. If clarity does not reflect chemistry, then something else has to be keeping aquariums stable, and that something is biology. Specifically, microbial biology. One of the biggest misunderstandings in fish keeping is where beneficial bacteria actually live, because they do not float freely in the water column in meaningful numbers, they colonize surfaces. Anything solid in an aquarium will then become a biological real estate. Filter media, substrate, glass, hardscapes, plant leaves. These surfaces develop biofilms, which are these thin, complex layers of bacteria, archaea, fungi, and microorganisms embedded in a matrix they produce themselves. This is where biological filtration will happen. The water column, by comparison, is relatively empty. Clear water often means that there is very little life suspended in it. That might look clean, but biologically it's almost irrelevant. What matters is what's attached. Peripyton layers on rocks and plants host nitrifying bacteria, but also a wide range of microfauna, protozoa, rotifers, tiny crustaceans. These microorganisms consume waste, regulate bacterial populations, and recycle nutrients in ways mechanical filtration cannot replicate. When systems are allowed to mature, these biological layers become stable. They buffer chemical fluctuations and they process waste continuously. They create redundancy, but they don't look polished. A tank with visible biofilm, light algae, or surface growth often has far more biological capacity than one that looks sterile. And this is what's called the clarity paradox. When aquariums are overfiltered, overcleaned, or aggressively polished, these biological layers are repeatedly removed or disrupted. The water will still look perfect, but the system will lose its resilience. There is no buffer when chemistry shifts. This is why some algae-free tanks crash unexpectedly. Not because algae was bad, but because the system lacked biological depth. Mild turbidity can also be functional. Slight bacterial presence in the water column, especially in mature systems, can be parts of nutrient processing. Not all cloudiness is a failure state. Sometimes it's transitional. Sometimes it's biological activity stabilizing itself. By contrast, sterile systems rely entirely on constant intervention. The moment maintenance slips or condition change, there is nothing in place to absorb the impact. So while clarity suggests control, biology requires complexity, and complexity rarely looks clean. Once you do understand how chemistry and biology actually work in an aquarium, a pattern will start to emerge. Many system failures don't happen in tanks that look neglected. They will often actually happen in tanks that looks perfect. One of the most common triggers is overcleaning. When surfaces are scrubbed too aggressively, filler media is rinsed excessively, or substrate is vacuumed down to bare material, biofilms will then be stripped away. The system loses its biological capacity all at once. Visually, the result will be very impressive. The water will look clear and the tank will generally look pristine. Biologically, you will have reset the system. With less surface biology available, waste processing slows, ammonia spikes become more likely, small changes that were previously buffered will now propagate through the system. The tank essentially becomes chemically fragile. Excessive mechanical filtration creates a similar problem. Highly polished water can give the impression of stability, but mechanical filtration removes particles and not dissolve the waste. If biological capacity do not scale with stocking and feeding, clarity masks accumulation rather than preventing it. Chemical water clarifiers push this even further. These products will bind fine particles together so they can be mechanically removed. The water will clear rapidly, but nothing about their underlying chemistry will change. Ammonia, nitrite, and nitrate will remain exactly where they are. So the symptom will disappear, but the cause will then still remain. New tank syndrome is another classic example. Fresh setups often look their best in the first few weeks. Clear water, clean surfaces, minimal growth, but the biological system has not matured. Microbial diversity is very low and the buffering is weak. When waste production increases or when a parameter shifts slightly, there is no depth to absorb it. The crash will feel sudden, but chemically it was inevitable. Low diversity systems behave the same way. Tanks that rely on minimal surface area, frequent sterilization, or constant intervention often run well until they don't. There's no redundancy, there's no biological backup. This is why some aquariums experience recurring fish loss despite looking flawless. The water will be clear and again the decor is spotless, but the system will lack stability. So take this away. Clarity will create confidence and stability creates longevity. When clarity is prioritized without biological support, the systems becomes brittle. It works only as long as conditions remain perfect, and aquariums never stay perfect. This is the point where clear water stops being a goal and starts becoming a signal. It's not always a problem, but it is something that needs context. Because in a functioning ecosystem, health is not defined by how little is visible, but by how much the system can tolerate without failing. Clarity without stability can then become a sign of fragility. So what will all this mean for your tanks? Just because the water is clear and the surfaces are polished doesn't mean that the aquarium is healthy. Those things tell you almost nothing about what's really happening chemically or biologically. Health comes from the invisible systems running under the surface. Bacteria on rocks and filter media, microfauna in the substrate, and the chemical balance in the water. If those systems are stable, your fish will thrive, even if the water isn't perfectly polished. If they're fragile, the tank can fail, even if it looks perfect. So next time you check a tank, don't rely on what you see. Look at the measurable indicators and consider how the system is functioning, not just how it appears. The science behind aquariums rarely actually do match with what we can see through the glass. Thank you for watching this episode of The Science Behind. If you've enjoyed the video and would like to see more, please do consider liking the video and subscribing. And also, let me know in the comments if you've ever seen the water in your aquarium becoming unclear, and also how you responded. It might be helpful to other people. I also do dabble in aquascaping, so if you want to see me making some attempts at that, you can find more videos on the channel. Bye for now.